Welcome back to G Fun Facts Online. For centuries, the scientific community approached the inner lives of animals with a profound skepticism, rooted in Cartesian philosophy, which famously reduced animals to mere biological automatons, reacting blindly to stimuli. The study of animal behavior spent much of the 20th century under the strict purview of behaviorism. In this era, attributing emotions to animals was dismissed as unscientific anthropomorphism. Fast forward to the present day, and we are in the midst of a spectacular paradigm shift. We have entered the era of animal affective neuroscience, a multidisciplinary field that not only acknowledges the rich, complex emotional lives of non-human animals, but actively seeks to map, measure, and understand them. Let us dive right into how science is achieving this. Historically, the study of animal welfare was profoundly negative in its focus. The benchmark for a good environment for a captive animal, whether on a farm, in a laboratory, or within a zoological park, was simply the absence of suffering. If an animal was not diseased, starving, or exhibiting severe signs of stress and fear, its welfare was considered adequate. Today, however, zoologists, ethologists, and neuroscientists agree that the mere absence of a negative state does not equate to a positive one. Just as a human life, devoid of pain, is not necessarily a happy one, an animal requires more than just survival to thrive. This realization has sparked a global scientific movement toward understanding and measuring positive animal emotions like joy, anticipation, pleasure, and playfulness, ushering in the concept of positive animal welfare and the pursuit of a good life for animals under human care. To understand how we measure positive emotions in animals today, we must first look to the foundational work of the late neuroscientist Jacques Panksepp, the undisputed father of affective neuroscience. Panksepp boldly challenged the behaviorist dogma by proving that mammals share deep, ancient neurological circuits responsible for generating primary emotional states. Through decades of mapping the subcortical regions of the mammalian brain areas that evolved long before the complex neocortex responsible for human reasoning, Panksepp identified 7 primary emotional operating systems. He capitalized these to distinguish them as fundamental neural networks. They are seeking rage, fear, lust, care, panic, grief, and play. While earlier welfare science fixated on mitigating fear and panic, modern positive zoology is intensely focused on the seeking and play systems. The seeking system is the brain's generalized reward and anticipation system, driven heavily by the neurotransmitter dopamine. It is the engine of curiosity, urging an animal to explore its environment, forage, and anticipate positive outcomes. When an animal is engaged in a puzzle feeder or sniffing out a new scent trail, its seeking system is activated, producing an energized, highly positive emotional state of eager anticipation. Then we have the play system, perhaps the most joyful of the primary circuits. It is hardwired into the mammalian brain to facilitate social bonding, physical coordination, and cognitive development. Panksepp famously demonstrated this by discovering rat laughter. When young rats engage in rough and tumble play, or when they are tickled by human researchers, they emit rapid, high-frequency 50 kHz ultrasonic vocalizations. These sounds, entirely inaudible to the human ear without specialized equipment, are the neurobiological equivalent of human giggling and represent a profound marker of positive affect. Furthermore, the work of Kent Berridge and others has expanded our understanding of positive affect by differentiating between wanting, which is incentive salience linked to dopamine, and the seeking system, and liking, which is hedonic pleasure linked to opioid and endocannabinoid hotspots in the brain. Measuring positive emotions in zoology today involves looking for behavioral and physiological markers of both the enthusiastic wanting and the blissful liking. The greatest challenge in animal affective neuroscience, however, is the lack of verbal self-reporting. An animal cannot simply fill out a psychological questionnaire to rate its happiness on a scale of 1 to 10. Therefore, scientists must rely on a sophisticated triad of indicators encompassing physiological markers, behavioral observations, and cognitive assessments. Let us explore the first pillar, physiological and neurological biomarkers. Historically, animal welfare was measured via cortisol levels, the quintessential stress hormone released by the adrenal glands. However, researchers are now looking beyond cortisol to identify the biochemical signatures of positive states. Advances in non-invasive technology are allowing researchers to peer directly into the working animal brain. Functional near-infrared spectroscopy, or FNIRS, is increasingly used to assess changes in cortical blood perfusion and neuronal activity associated with positive mood and emotional reactions in animals. Similarly, electroencephalography, or EEG patterns, are being decoded to distinguish between states of relaxed wakefulness, active engagement, and stress. In laboratory settings, microinjections into specific brain structures, such as the nucleus accumbens, have allowed researchers to map the exact neural substrates of sensory pleasure. We are also looking at hormonal and cellular indicators. Oxytocin, often dubbed the love hormone, is a powerful physiological indicator of positive social affect. Elevated oxytocin levels are observed in animals engaging in mutual grooming, maternal care, and affiliative behaviors, even across species, such as the profound bond between a dog and its human guardian. Endorphins and brain-derived neurotrophic factor, or BDNF, are also being studied as biological markers of a thriving animal. At a cellular level, researchers are even examining telomere attrition rates. Animals experiencing chronic positive welfare may show slower degradation of their telomeres compared to those in stressful environments. Furthermore, positive emotions cause subtle shifts in the autonomic nervous system. Heart rate variability. The variation in time between consecutive heartbeats is a highly sensitive measure. A higher heart rate variability is generally associated with parasympathetic dominance, the rest and digest or relaxed state, and is considered a strong physiological indicator of positive welfare. Thermal imaging has also revealed that emotional states affect peripheral blood flow. In dairy cows, for example, a drop in nasal temperature has been correlated with the experience of positive emotions, likely due to a relaxing of the sympathetic nervous system. Eye temperature variations are also being validated as non-invasive autonomic markers of emotional valence. The second pillar involves behavioral indicators of joy. While physiological measures require equipment and sometimes invasive sampling, behavioral indicators provide a continuous real-time window into an animal's emotional state. Identifying validated behavioral markers of positive affect is currently one of the most dynamic areas of zoological research. Across species from dairy calves to captive dolphins, the presence of play is universally recognized as the gold standard of positive animal welfare. Play only occurs when an animal's basic needs are met, because an animal that is hungry, terrified, or sick will not expend valuable energy on play. Solitary locomotor play, such as a horse frolicking in a pasture or a mink playfully attacking a wooden block, and social play like rough and tumble interactions, indicate that the animal feels secure and is experiencing endogenous opioid release. In zoo environments, the absence of stereotypic behaviors like repetitive pacing was once considered proof of good welfare. Today, zoologists measure behavioral diversity, which is the richness and evenness of an animal's behavioral repertoire. Using metrics like the Shannon Diversity Index, researchers have found a significant inverse relationship between high behavioral diversity and fecal glucocorticoid stress metabolites in species ranging from cheetahs to chimpanzees and bottlenose dolphins. A high diversity score suggests the animal is actively engaging with its environment, utilizing its seeking system and thriving. In herd and flock animals, behavioral synchrony, such as an entire group of cows choosing to lie down and ruminate at the exact same time, is a strong indicator of social harmony and positive group welfare. Beyond rat laughter, positive vocalizations exist across the entire animal kingdom, from the purring of felines to the complex melodic social calls of certain primates and birds. Body language also speaks volumes. In dairy cows and sheep, specific ear postures, such as relaxed backward-hanging ears, are highly correlated with positive experiences like being brushed or anticipating a food reward. In dogs, affiliative behaviors such as relaxed tail wagging, soliciting chase, and the iconic playbow are empirical evidence of a comfortable positive state. The third pillar, cognitive bias testing, often referred to as judgment bias, is one of the most elegant and revolutionary methods for assessing animal emotion. Derived from human psychology, this paradigm tests whether an animal is an optimist or a pessimist. The underlying principle is that an individual's background emotional state heavily influences how they interpret ambiguous information. In a typical animal judgment bias test, an animal is trained to associate one cue, such as a high-pitched tone or a bowl placed on the left side of a room, with a highly positive reward, like a favorite treat. They are then trained to associate a different cue, like a low-pitched tone or a bowl on the right, with a negative outcome, such as no treat or a mild puff of air. Once the animal has mastered this discrimination, the researchers introduce an ambiguous cue, a medium-pitched tone, or a bowl, placed exactly in the middle of the room. How the animal reacts to this ambiguous cue reveals its underlying mood. An animal experiencing chronic stress or poor welfare will typically demonstrate a pessimistic cognitive bias, treating the ambiguous cue as if it were the negative one, and avoiding it. Conversely, an animal living in an enriched environment that promotes positive affect will display an optimistic cognitive bias, eagerly approaching the ambiguous cue in the hope of a reward. This methodology has been successfully applied to dogs, pigs, sheep rats, and even honeybees, providing a robust quantifiable measure of positive emotional valence. The fourth and final pillar is qualitative behavior assessment, or QBA. While objective measurements like heart rate variability and cognitive bias are vital, researchers have also scientifically validated the power of structured human observation. QBA is a methodology that evaluates the expressive quality of an animal's movement and posture. Rather than simply counting how many times a dog wags its tail, QBA asks the observer to assess how the dog is behaving. Is it behaving in a way that is relaxed, sociable, playful, apathetic, or anxious? By utilizing standardized fixed lists of descriptive terms and analyzing the data through sophisticated statistical methods like generalized procrustes analysis, QBA has proven to be a highly reliable, rapid, and non-invasive tool for directly inferring an animal's emotional state. It captures the whole animal dynamic, integrating seamlessly into modern welfare assessment protocols on farms and in zoos. This massive shift toward measuring positive emotions has completely restructured the global frameworks we use to evaluate animal care. For decades, the gold standard was the 5 freedoms, such as freedom from hunger and freedom from pain. Today, the scientific consensus has moved to the 5 domains model. The 5 domains of nutrition, physical environment, health, and behavioral interactions all feed into the crucial 5th domain, which is mental state. This model explicitly recognizes that simply providing food and shelter is insufficient. Care providers must facilitate positive mental experiences. A central pillar to achieving a positive mental state is agency. Agency is the ability of an animal to make meaningful choices and exert control over its environment. Neuroscience shows us that the mammalian brain is intrinsically rewarded by the act of solving problems and achieving goals. A zoo animal that is simply handed food experiences basic satiation. However, an animal that must solve a complex puzzle, navigate a maze, or use tools to acquire its food engages its seeking system. The successful execution of this task triggers a surge of endogenous rewards, fostering competence, resilience, and a profound sense of positive welfare. Modern zoological habitat design now prioritizes dynamic, unpredictable environments that give animals choices, whether it is choosing which companion to sleep next to which microclimate to rest in, or whether to remain visible to the public or retreat to a private sanctuary. The application of affective neuroscience is not limited to mammals either. As our tools for measuring positive affect grow more refined, we are uncovering the emotional capacities of a staggering array of species. In agricultural science, there is a massive push to validate positive welfare indicators to improve the lives of livestock. Research on dairy cows has shown that access to pasture, the ability to explore, and the synchronization of resting behaviors are profound indicators of positive welfare. When cows are granted the agency to express their natural behavioral repertoire, their physical health improves, and they display clear markers of positive affect, including relaxed ear postures and increased social grooming. For companion animals like dogs and cats, the home environment can sometimes be as restrictive as a cage if not properly enriched. Affective neuroscience has highlighted that individual variation, such as a dog's inherent personality attachment style and temperamental shy boldness, dictates how they experience the world. Providing companion animals with diverse behavioral interactions training that utilizes positive reinforcement, which activates the seeking system, and ample opportunities for play are scientifically proven methods to ensure they live a good life. Most astonishingly, the search for positive emotions is pushing the boundaries of taxonomy. Fish have been observed engaging in complex play behaviors and seeking out tactile stimulation, like being gently stroked by a diver, or rubbing against a preferred texture, which lowers their physiological stress markers. Even invertebrates are entering the conversation. Recent groundbreaking studies on bumblebees have demonstrated that they will go out of their way to roll small wooden balls, a behavior that provides no survival or foraging benefit. The bees roll the balls repeatedly, and younger bees play more than older ones, which is a classic hallmark of mammalian play behavior. This suggests that the neurological capacity for fun and positive affective states may be an ancient, deeply conserved evolutionary trait stretching far beyond the mammalian lineage. The explosion of data from animal affective neuroscience carries profound ethical implications. Acknowledging that animals from the cows in our pastures to the elephants in our zoos, and perhaps even the bees in our gardens, are sentient beings capable of experiencing profound joy, anticipation, and pleasure completely alters the moral landscape of human-animal interactions. We are moving away from an era of paternalistic care, where humans dictated every aspect of an animal's sterile existence, to ensure physical health into an era of collaborative coexistence. Zoos are transforming from living museums into dynamic environments engineered for emotional flourishing. Agricultural systems face increasing scientific and public pressure to drastically alter husbandry practices to allow for the expression of natural behaviors, exploration, and play. Looking to the future, the integration of artificial intelligence and machine learning will revolutionize our ability to measure positive emotions. AI algorithms are currently being trained to monitor 24/7 video feeds of zoo and farm animals to automatically calculate behavioral diversity, track complex social interactions, and identify micro-expressions of relaxation or joy that a human observer might miss. Bioacoustical monitoring networks can continuously record and decode animal vocalizations, providing real-time dashboards of the emotional valence of a flock herd or individual. Furthermore, as the technology for portable non-invasive neuroimaging like wireless EEG and FNIRS becomes more robust, we will soon be able to directly monitor the brain activity of free-moving animals in complex environments, mapping the exact moments when an animal experiences a surge of happiness. In conclusion, animal affective neuroscience has forever shattered the illusion of the unfeeling animal machine. By rigorously measuring the physiological, behavioral, and cognitive dimensions of positive emotions, science is proving what pet owners and animal lovers have intuitively known for millennia. Animals feel deeply. As we continue to refine our metrics for joy, play, and anticipation in the animal kingdom, we are not just advancing zoological science, we are elevating our own humanity, stepping into a future where the deliberate cultivation of animal happiness is considered one of our highest moral imperatives. Thank you for tuning in to G Fun Facts Online, where we bring you the most fascinating discoveries from the frontiers of science.